highest possible degree that natural selection could produce such an instrument in gradual steps. Even so, and to the unending exasperation of his supporters, Darwin not only insisted that all change was gradual, but in nearly every edition of Origin stepped up the amount of time he supposed necessary to allow evolution to progress, which pushed his ideas increasingly out of favor. Eventually, according to the scientist and historian Jeffrey Schwartz, Darwin lost virtually all the support that still remained among the ranks of fellow natural historians and geologists. Ironically, considering that Darwin called his book On the Origin of Species, the one thing he couldn't explain was how species originated. Darwin's theory suggested a mechanism for how a species might become stronger or better or faster, in a word, fitter, but gave no indication of how it might throw up a new species. A Scottish engineer, Fleming Jenkin, considered the problem and noted an important flaw in Darwin's argument. Darwin believed that any beneficial trait that arose in one generation would be passed on to subsequent generations, thus strengthening the species. Jenkin pointed out that a favorable trait in one parent wouldn't become dominant in succeeding generations, but in fact would be diluted through blending. If you pour whiskey into a tumbler of water, you don't make the whiskey stronger, you make it weaker. And if you pour that dilute solution into another glass of water, it becomes weaker still. In the same way, any favorable trait introduced by one parent would be successively watered down by subsequent matings until it ceased to be apparent at all. Thus, Darwin's theory was a recipe not for change, but for constancy. Lucky flukes might arise from time to time, but they would soon vanish under the general impulse to bring everything back to a stable mediocrity. If natural selection were to work, some alternative, unconsidered mechanism was required. Unknown to Darwin and to everyone else, 1,200 kilometers away in a tranquil corner of Middle Europe, a retiring monk named Gregor Mendel was coming up with a solution. Mendel was born in 1822 to a humble farming family in a backwater of the Austrian Empire in what is now the Czech Republic. Schoolbooks once portrayed him as a simple but observant provincial monk whose discoveries were largely serendipitous, the result of noticing some interesting traits of inheritance while pottering about with pea plants in the monastery's kitchen garden. In fact, Mendel was a trained scientist. He had studied physics and mathematics at the Olmütz Philosophical Institute and University of Vienna, and he brought scientific discipline to all he did. Moreover, the monastery at Brno, where he lived from 1843, was known as a learned institution. It had a library of 20,000 books and a tradition of careful scientific investigation. Before embarking on his experiments, Mendel spent 2 years preparing his control specimens, 7 varieties of pea, to make sure they bred true. Then, helped by 2 full-time assistants, he repeatedly bred and crossbred hybrids from 30,000 pea plants. It was delicate work, requiring the 3 men to take the most exacting pains to avoid accidental cross-fertilization and to note every slight variation in the growth and appearance of seeds, pods, leaves, stems, and flowers. Mendel knew what he was doing. He never used the word gene. It wasn't coined until 1913 in an English medical dictionary, though he did invent the terms dominant and recessive. What he established was that every seed contained 2 factors, or elementa, as he called them, a dominant one and a recessive one. And these factors, when combined, produced predictable patterns of inheritance. The results he converted into precise mathematical formulae. Altogether, Mendel spent 8 years on the experiments, then confirmed his results with similar experiments on flowers, corn, and other plants. If anything, Mendel was too scientific in his approach, for when he presented his findings at the February and March meetings of the Natural History Society of Brno in 1865, the audience of about 40 listened politely, but was conspicuously unmoved, even though the breeding of plants was a matter of great practical interest to many of the members. When Mendel's report was published, he eagerly sent a copy to the great Swiss botanist Carl Wilhelm von Nageli, whose support was more or less vital for the theory's prospects. Unfortunately, Nageli failed to perceive the importance of what Mendel had found. He suggested that Mendel try breeding hawkweed. Mendel dutifully obeyed, but quickly realized that hawkweed had none of the requisite features for studying heritability. It was evident that Nageli had not read the paper closely or possibly at all. Frustrated, Mendel retired from investigating heritability and spent the rest of his life growing outstanding vegetables and studying bees, mice, and sunspots, among much else. Eventually, he was made abbot. Mendel's findings weren't quite as widely ignored as is sometimes suggested. His study received a glowing entry in the Encyclopaedia Britannica, then a more leading record of scientific thought than now, and was cited repeatedly in an important paper by the German Wilhelm Olbers-Focke. Indeed, it was because Mendel's ideas never entirely sank below the waterline of scientific thought that they were so easily recovered when the world was ready for them. Together, without realizing it, Darwin and Mendel laid the groundwork for all of life sciences in the 20th century. Darwin saw that all living things are connected, that ultimately they trace their ancestry to a single common source. Mendel's work provided the mechanism to explain how that could happen. The two men could easily have helped each other. Mendel owned a German edition of The Origin of Species, which he is known to have read, so he must have realized the applicability of his work to Darwin's. Yet he appears to have made no effort to get in touch. And Darwin, for his part, is known to have studied Focke's influential paper with its repeated references to Mendel's work, but didn't connect them to his own studies. The one thing everyone thinks featured in Darwin's argument, that humans are descended from apes, didn't feature at all, except as one passing allusion. Even so, it took no great leap of imagination to see the implications for human development in Darwin's theories, and it became an immediate talking point. The showdown came on Saturday, the 30th of June, 1860, at a meeting of the British Association for the Advancement of Science in Oxford. Huxley had been urged to attend by Robert Chambers, author of Vestiges of the Natural History of Creation, though he was still unaware of Chambers's connection to that contentious tome. Darwin, as ever, was absent. The meeting was held at the Oxford Zoological Museum. More than 1,000 people crowded into the chamber. Hundreds more were turned away. People knew that something big was going to happen, though they had first to wait while a slumber-inducing speaker named John William Draper of New York University bravely slogged his way through 2 hours of introductory remarks on the intellectual development of Europe considered with reference to the views of Mr. Darwin. Finally, the Bishop of Oxford, Samuel Wilberforce, rose to speak. Wilberforce had been briefed, or so it is generally assumed, by the ardent anti-Darwinian Richard Owen, who had been a guest in his home the night before. As nearly always with events that end in uproar, accounts of what exactly transpired vary widely. In the most popular version, Wilberforce, when properly in flow, turned to Huxley with a dry smile and demanded of him whether he claimed attachment to the apes by way of his grandmother or grandfather. The remark was doubtless intended as a quip, but it came across as an icy challenge. According to his own account, Huxley turned to his neighbor and whispered, "The Lord hath delivered him into my hands," then rose with a certain relish. Others, however, recalled a Huxley trembling with fury and indignation. At all events, Huxley declared that he would rather claim kinship to an ape than to someone who used his eminence to propound uninformed twaddle in what was supposed to be a serious scientific forum. Such a riposte was scandalous impertinence, as well as an insult to Wilberforce's office, and the proceedings instantly collapsed into tumult. A Lady Brewster fainted. Robert Fitzroy, Darwin's companion on the Beagle 25 years before, wandered through the hall with a Bible held aloft, shouting, "The book! The book!" He was at the conference to present a paper on storms in his capacity as head of the newly created meteorological department. Interestingly, each side afterwards claimed to have routed the other. Darwin did eventually make his belief in our kinship with the apes explicit in The Descent of Man in 1871. The conclusion was a bold one, since nothing in the fossil record supported such a notion. The only known early human remains of that time were the famous Neanderthal bones from Germany and a few uncertain fragments of jaw bones, and many respected authorities refused to believe even in their antiquity. The Descent of Man was altogether a more controversial book than The Origin, but by the time of its appearance the world had grown less excitable, and its arguments caused much less of a stir. For the most part, however, Darwin passed his twilight years on other projects, most of which touched only tangentially on questions of natural selection. He spent amazingly long periods picking through bird droppings, scrutinizing the contents in an attempt to understand how seeds spread between continents, and spent years more studying the behavior of worms. One of his experiments was to play the piano to them, not to amuse them, but to study the effects on them of sound and vibration. He was the first to realize how vitally important worms are to soil fertility. It may be doubted whether there are many other animals which have played so important a part in the history of the world. He wrote in his masterwork on the subject, The Formation of Vegetable Mold Through the Action of Worms, 1881, which was actually more popular than On the Origin of Species had ever been. Among his other books were On the Various Contrivances by which British and Foreign Orchids are Fertilized by Insects, 1862, Expressions of the Emotions in Man and Animals, 1872, which sold almost 5,300 copies on its first day, The Effects of Cross and Self-Fertilization in the Vegetable Kingdom, 1876, a subject that came improbably close to Mendel's own work without attaining anything like the same insights, and The Power of Movement in Plants. Finally, but not least, he devoted much effort to studying the consequences of inbreeding, a matter of private interest to him. Having married his own cousin, Darwin glumly suspected that certain physical and mental frailties among his children arose from a lack of diversity in his family tree. Darwin was often honored in his lifetime, but never for On the Origin of Species or The Descent of Man. When the Royal Society bestowed on him the prestigious Copley Medal, it was for his geology, zoology, and botany, not evolutionary theories. And the Linnaean Society was similarly pleased to honor Darwin without embracing his radical notions. He was never knighted, though he was buried in Westminster Abbey next to Newton. He died at Down in April 1882. Mendel died 2 years later. Darwin's theory didn't really gain widespread acceptance until the 1930s and 1940s, with the advance of a refined theory called, with a certain hauteur, the modern synthesis, combining Darwin's ideas with those of Mendel and others. For Mendel, appreciation was also posthumous, though it came somewhat sooner. In 1900, 3 scientists working separately in Europe rediscovered Mendel's work more or less simultaneously. It was only because one of them, a Dutchman named Hugo de Vries, seemed set to claim Mendel's insights as his own, that a rival made it noisily clear that the credit really lay with the forgotten monk. The world was almost, but not quite, ready to begin to understand how we got here, how we made each other. It is fairly amazing to reflect that at the beginning of the 20th century, and for some years beyond, the best scientific minds in the world couldn't actually tell you in any meaningful way where babies came from. And these, you may recall, were men who thought science was nearly at an end. Chapter 26, The Stuff of Life. If your 2 parents hadn't bonded just when they did, possibly to the second, possibly to the nanosecond, you wouldn't be here. And if their parents hadn't bonded in a precisely timely manner, you wouldn't be here either. And if their parents hadn't done likewise, and their parents before them, and so on, obviously and indefinitely, you wouldn't be here. Push backwards through time, and these ancestral debts begin to add up. Go back just 8 generations to about the time that Charles Darwin and Abraham Lincoln were born, and already there are over 250 people on whose timely couplings your existence depends. Continue further to the time of Shakespeare and the Mayflower Pilgrims, and you have no fewer than 16,384 ancestors, earnestly exchanging genetic material in a way that would eventually and miraculously result in you. At 20 generations ago, the number of people procreating on your behalf has risen to 1,048,576. 5 generations before that, and there are no fewer than 33,554,432 men and women on whose devoted couplings your existence depends. By 30 generations ago, your total number of forebears, remember these aren't cousins and aunts and other incidental relatives, but only parents and parents of parents in a line leading ineluctably to you, is over 1 billion. 1,073,741,824, to be precise. If you go back 64 generations to the time of the Romans, the number of people on whose cooperative efforts your eventual existence depends has risen to approximately 1 million trillion, which is several thousand times the total number of people who have ever lived. Clearly, something has gone wrong with our maths here. The answer, it may interest you to learn, is that your line is not pure. You couldn't be here without a little incest. Actually, quite a lot of incest, albeit at a genetically discrete remove. With so many millions of ancestors in your background, there will have been many occasions when a relative from your mother's side of the family procreated with some distant cousin from your father's side of the ledger. In fact, if you are in a partnership now with someone from your own race and country, the chances are excellent that you are at some level related. Indeed, if you look around you on a bus or in a park or cafe or any crowded place, most of the people you see are very probably relatives. When someone boasts to you that he is descended from Shakespeare or William the Conqueror, you should answer at once, "Me too." In the most literal and fundamental sense, we are all family. We are also uncannily alike. Compare your genes with any other human beings, and on average there will be about 99.9% the same. That is what makes us a species. The tiny differences in that remaining 0.1%, roughly 1 nucleotide base in every 1,000, to quote the British geneticist and recent Nobel laureate John Sulston, are what endow us with our individuality. Much has been made in recent years of the piecing together of the human genome. In fact, there is no such thing as the human genome. Every human genome is different. Otherwise, we would all be identical. It is the endless recombinations of our genomes, each nearly identical to all the others, but not quite, that make us what we are, both as individuals and as a species. But what exactly is this thing we call the genome? And what, come to that, are genes? Well, start with a cell again. Inside the cell is a nucleus, and inside each nucleus are the chromosomes, 46 little bundles of complexity, of which 23 come from your mother and 23 from your father. With a very few exceptions, every cell in your body, 99.999% of them say, carries the same complement of chromosomes. The exceptions are the red blood cells, some immune system cells, and egg and sperm cells, which for various organizational reasons don't carry the full genetic package. Chromosomes constitute the complete set of instructions necessary to make and maintain you, and are made of long strands of a little wonder chemical called deoxyribonucleic acid, or DNA, the most extraordinary molecule on Earth, as it has been called. DNA exists for just one reason, to create more DNA. And you have a lot of it inside you, nearly 2 meters of it squeezed into almost every cell. Each length of DNA comprises some 3.2 billion letters of coding, enough to provide 10 to the 3,480 millionth possible combinations, guaranteed to be unique against all conceivable odds, in the words of Christian de Duve. That's a lot of possibility. A 1 followed by more than 3 billion zeros. It would take more than 5,000 average-sized books just to print that figure, notes Deduve. Look at yourself in the mirror and reflect upon the fact that you are beholding 10,000 trillion cells and that almost every one of them holds 2 yards of densely compacted DNA, and you begin to appreciate just how much of this stuff you carry around with you. If all your DNA were woven into a single fine strand, there would be enough of it to stretch from the Earth to the Moon and back, not once or twice, but again and again. Altogether, according to one calculation, you may have as much as 20 million kilometers of DNA bundled up inside you. Your body, in short, loves to make DNA, and without it, you couldn't live. Yet DNA is not itself alive. No molecule is, but DNA is, as it were, especially unalive. It is among the most non-reactive, chemically inert molecules in the living world, in the words of the geneticist Richard Lewontin. That is why it can be recovered from patches of long-dried blood or semen in murder investigations and coaxed from the bones of ancient Neanderthals. It also explains why it took scientists so long to work out how a substance so mystifyingly low-key, so in a word, lifeless, could be at the very heart of life itself. As a known entity, DNA has been around longer than you might think. It was discovered as far back as 1869 by Johann Friedrich MÉCH, a Swiss scientist working at the University of Tübingen in Germany. While delving microscopically through the pus in surgical bandages, MÉCH found a substance he didn't recognize and called it nuclein because it resided in the nuclei of cells. At the time, MÉCH did little more than note its existence, but nuclein clearly remained on his mind. For 23 years later, in a letter to his uncle, he raised the possibility that such molecules could be the agents behind heredity. This was an extraordinary insight, but one so far in advance of the day's scientific requirements that it attracted no attention at all. For most of the next half-century, the common assumption was that the material, now called deoxyribonucleic acid, or DNA, had at most a subsidiary role in matters of heredity. It was too simple. It had just 4 basic components called nucleotides, which was like having an alphabet of just 4 letters. How could you possibly write the story of life with such a rudimentary alphabet? The answer is that you do it in much the way that you create complex messages with the simple dots and dashes of Morse code, by combining them. DNA didn't do anything at all, as far as anyone could tell. It just sat there in the nucleus, possibly binding the chromosome in some way, or adding a splash of acidity on command, or fulfilling some other trivial task that no one had yet thought of. The necessary complexity, it was thought, had to exist in proteins in the nucleus. There were, however, 2 problems with dismissing DNA. First, there was so much of it, nearly 2 meters in nearly every nucleus, so clearly the cells esteemed it in some important way. On top of this, it kept turning up like the suspect in a murder mystery in experiments. In 2 studies in particular, one involving the pneumococcus bacterium and another involving bacteriophages, viruses that infect bacteria, DNA betrayed an importance that could be explained only if its role were more central than prevailing thought allowed. The evidence suggested that DNA was somehow involved in the making of proteins, a process vital to life. Yet it was also clear that proteins were being made outside the nucleus, well away from the DNA that was supposedly directing their assembly. No one could understand how DNA could possibly be getting messages to the proteins. The answer, we now know, was RNA, or ribonucleic acid, which acts as an interpreter between the 2. It is a notable oddity of biology that DNA and proteins don't speak the same language. For almost 4 billion years, they have been living the world's great double act, and yet they answer to mutually incompatible codes, as if one spoke Spanish and the other Hindi. To communicate, they need a mediator in the form of RNA. Working with a kind of chemical clerk called a ribosome, RNA translates information from a cell's DNA into terms proteins can understand and act upon. However, by the early 1900s, where we resume our story, we were still a very long way from understanding that, or indeed almost anything else to do with the confused business of heredity. Clearly, there was a need for some inspired and clever experimentation. And happily, the age produced a young person with a diligence and aptitude to undertake it. His name was Thomas Hunt Morgan, and in 1904, just 4 years after the timely rediscovery of Mendel's experiments with pea plants, and still almost a decade before gene would even become a word, he began to do remarkably dedicated things with chromosomes. Chromosomes had been discovered by chance in 1888, and were so called because they readily absorbed dye, and thus were easy to see under the microscope. By the turn of the century, it was strongly suspected that they were involved in the passing on of traits, but no one knew how, or even really whether they did this. Morgan chose as his subject of study a tiny, delicate fly formerly called Drosophila melanogaster, but more commonly known as the fruit fly, or vinegar fly, banana fly, or garbage fly. Drosophila is familiar to most of us as that frail, colorless insect that seems to have a compulsive urge to drown in our drinks. As laboratory specimens, fruit flies had certain very attractive advantages. They cost almost nothing to house and feed, could be bred by the millions in milk bottles, went from egg to productive parenthood in 10 days or less, and had just 4 chromosomes, which kept things conveniently simple. Working out of a small lab, which became known inevitably as the Fly Room, in Schermerhorn Hall at Columbia University in New York, Morgan and his team embarked on a program of meticulous breeding and crossbreeding involving millions of flies. One biographer says billions, though that is probably an exaggeration, each of which had to be captured with tweezers and examined under a jeweler's glass for any tiny variations in inheritance. For 6 years, they tried to produce mutations by any means they could think of, zapping the flies with radiation and X-rays, rearing them in bright light and darkness, baking them gently in ovens, spinning them crazily in centrifuges, but nothing worked. Morgan was on the brink of giving up when there occurred a sudden and repeatable mutation, a fly that had white eyes rather than the usual red ones. With this breakthrough, Morgan and his assistants were able to generate useful deformities, allowing them to track a trait through successive generations. By such means, they could work out the correlations between particular characteristics and individual chromosomes, eventually proving to more or less everyone's satisfaction that chromosomes were at the heart of inheritance. The problem, however, remained the next level of biological intricacy, the enigmatic genes and the DNA that composed them. These were much trickier to isolate and understand. As late as 1933, when Morgan was awarded a Nobel Prize for his work, many researchers still weren't convinced that genes even existed. As Morgan noted at the time, there was no consensus as to what the genes are, whether they are real or purely fictitious. It may seem surprising that scientists could struggle to accept the physical reality of something so fundamental to cellular activity. But as Wallace, King, and Sanders point out in Biology, the Science of Life, that rarest thing, a readable textbook, we are in much the same position today in respect of mental processes such as thought and memory. We know that we have them, of course, but we don't know what, if any, physical form they take. So it was for a very long time with genes. The idea that you could pluck one from your body and take it away for study was as absurd to many of Morgan's peers as the idea that scientists today might capture a stray thought and examine it under a microscope. What was certainly true was that something associated with chromosomes was directing cell replication. Finally, in 1944, after 15 years of effort, a team at the Rockefeller Institute in Manhattan, led by a brilliant but diffident Canadian named Oswald Avery, succeeded with an exceedingly tricky experiment in which an innocuous strain of bacteria was made permanently infectious by crossing it with alien DNA, proving that DNA was far more than a passive molecule and almost certainly was the active agent in heredity. The Austrian-born biochemist Erwin Chargaff later quite seriously suggested that Avery's discovery was worth 2 Nobel Prizes. Unfortunately, Avery was opposed by one of his own colleagues at the Institute, a strong-willed and disagreeable protein enthusiast named Alfred Mirsky, who did everything in his power to discredit Avery's work, including, it has been said, lobbying the authorities at the Karolinska Institute in Stockholm not to give Avery a Nobel Prize. Avery by this time was 66 years old and tired. Unable to deal with the stress and controversy, he resigned his position and never went near a lab again. But other experiments elsewhere overwhelmingly supported his conclusions, and soon the race was on to find the structure of DNA. Had you been a betting person in the early 1950s, your money would almost certainly have been on Linus Pauling of Caltech, America's leading chemist, to crack the structure of DNA. Pauling was unrivaled in determining the architecture of molecules and had been a pioneer in the field of X-ray crystallography, the technique that would prove crucial to peering into the heart of DNA. In an exceedingly distinguished career, he would win 2 Nobel Prizes, for chemistry in 1954 and peace in 1962. But with DNA, he became convinced that the structure was a triple helix, not a double one, and never quite got on the right track. Instead, victory fell to an unlikely quartet of scientists in England, who didn't work as a team, often weren't on speaking terms, and were for the most part novices in the field. Of the 4, the nearest to a conventional boffin was Maurice Wilkins, who had spent much of the Second World War helping to design the atomic bomb. 2 of the others, Rosalind Franklin and Francis Crick, had spent their war years working for the British government on mines, Crick on the type that blow up, Franklin on the type that produce coal. The most unconventional of the foursome was James Watson, an American prodigy who as a boy had distinguished himself as a member of a highly popular radio program called The Quiz Kids, and thus could claim to be at least part of the inspiration for some of the members of the Glass family in Franny and Zooey and other works by J.D. Salinger, and who had entered the University of Chicago aged just 15. He had earned his PhD by the age of 22 and was now attached to the famous Cavendish Laboratory in Cambridge. In 1951, he was a gawky 23-year-old with a strikingly lively head of hair that appears in photographs to be straining to attach itself to some powerful magnet just out of frame. Crick, 12 years older and still without a doctorate, was less memorably hirsute and slightly more tweedy. In Watson's account, he is presented as blustery, nosy, cheerfully argumentative, impatient with anyone slow to share a notion, and constantly in danger of being asked to go elsewhere. Neither was formally trained in biochemistry. They assumed, correctly as it turned out, that if you could determine the shape of a DNA molecule, you would be able to see how it did what it did. They hoped to achieve this, it would appear, by doing as little work as possible beyond thinking, and no more of that than was absolutely necessary. As Watson cheerfully, if not a touch disingenuously, remarked in his autobiographical book, The Double Helix, "It was my hope that the gene might be solved without my learning any chemistry." They weren't actually assigned to work on DNA, and at one point were ordered to stop doing it. Watson was ostensibly mastering the art of crystallography. Crick was supposed to be completing a thesis on the X-ray diffraction of large molecules. Although Crick and Watson enjoy nearly all the credit in popular accounts for solving the mystery of DNA, their breakthrough was crucially dependent on experimental work done by their competitors, the results of which were obtained fortuitously, in the tactful words of the historian Lisa Jardine. Far ahead of them, at least at the beginning, were 2 academics at King's College in London, Wilkins and Franklin. The New Zealand-born Wilkins was a retiring figure, almost to the point of invisibility. A 1998 PBS documentary on the discovery of the structure of DNA, a feat for which he shared the 1962 Nobel Prize with Crick and Watson, managed to overlook him entirely. Franklin was the most enigmatic character of them all. In a severely unflattering portrait, Watson in the double helix depicted Franklin as a woman who was unreasonable, secretive, chronically uncooperative, and, this seemed especially to irritate him, almost willfully unsexy. He allowed that she was not unattractive and might have been quite stunning had she taken even a mild interest in clothes. But in this, she disappointed all expectations. She didn't even use lipstick, he noted in wonder, while her dress sense showed all the imagination of English blue-stocking adolescents. However, she did have the best images in existence of the possible structure of DNA, achieved by means of X-ray crystallography, the technique perfected by Linus Pauling. Crystallography had been used successfully to map atoms and crystals, hence crystallography, but DNA molecules were a much more finicky proposition. Only Franklin was managing to get good results from the process, but to Wilkins's perennial exasperation, she refused to share her findings. If Franklin was not warmly forthcoming with her findings, she cannot be altogether blamed. Female academics at King's in the 1950s were treated with a formalized disdain that dazzles modern sensibilities, actually any sensibilities. However senior or accomplished, they were not allowed into the college's senior common room, but instead had to take their meals in a more utilitarian chamber that even Watson conceded was dingily poky. On top of this, she was being constantly pressed, at times actively harassed, to share her results with a trio of men whose desperation to get a peek at them was seldom matched by more engaging qualities like respect. I'm afraid we always used to adopt, let's say, a patronizing attitude towards her, Crick later recalled. Two of these men were from a competing institution, and the third was more or less openly siding with them. It should hardly come as a surprise that she kept her results locked away. That Wilkins and Franklin did not get along was a fact that Watson and Crick seemed to have exploited to their benefit. Although the two of them were trespassing rather unashamedly on Wilkins's territory, it was with them that he increasingly sided, not altogether surprisingly, since Franklin herself was beginning to act in a decidedly queer way. Although her results showed that DNA definitely was helical in shape, she insisted to all that it was not. To Wilkins's presumed dismay and embarrassment, in the summer of 1952, she posted a mock notice around the King's physics department that said, "It is with great regret that we have to announce the death on Friday the 18th of July, 1952, of DNA helix. It is hoped that Dr. MHF Wilkins will speak in memory of the late helix." The outcome of all this was that in January 1953, Wilkins showed Watson Franklin's images, apparently without her knowledge or consent. It would be an understatement to call it a significant help to him. Years later, Watson conceded that it was the key event. It mobilized us. Armed with a knowledge of the DNA molecule's basic shape and some important elements of its dimensions, Watson and Crick redoubled their efforts. Everything now seemed to go their way. At one point, Pauling was en route to a conference in England, at which he would in all likelihood have met Wilkins and learned enough to correct the misconceptions that had put him on the wrong line of inquiry. But this was the McCarthy era, and Pauling found himself detained at Idlewild Airport in New York, his passport confiscated, on the grounds that he was too liberal of temperament to be allowed to travel abroad. Crick and Watson also had the no less convenient good fortune that Pauling's son was working at the Cavendish and innocently kept them abreast of any news of developments and setbacks at home. Still facing the possibility of being trumped at any moment, Watson and Crick applied themselves feverishly to the problem. It was known that DNA had 4 chemical components called adenine, guanine, cytosine, and thymine, and that these paired up in particular ways. By playing with pieces of cardboard cut into the shape of molecules, Watson and Crick were able to work out how the pieces fit together. From this, they made a mechano-like model, perhaps the most famous in modern science, consisting of metal plates bolted together in a spiral, and invited Wilkins, Franklin, and the rest of the world to have a look. Any informed person could see at once that they had solved the problem. It was, without question, a brilliant piece of detective work, with or without the boost of Franklin's picture. The 25th of April 1953 edition of Nature carried a 900-word article by Watson and Crick titled "A Structure for Deoxyribose Nucleic Acid." Accompanying it were separate articles by Wilkins and Franklin. It was an eventful time in the world. Edmund Hillary was just about to clamber to the top of Everest, while Elizabeth II was shortly to be crowned queen. So the discovery of the secret of life was mostly overlooked. It received a small mention in the News Chronicle and was ignored elsewhere. Rosalind Franklin did not share in the Nobel Prize. She died of ovarian cancer at the age of just 37 in 1958, 4 years before the award was granted. Nobel Prizes are not awarded posthumously. The cancer almost certainly arose as a result of chronic overexposure to X-rays through her work and could have been avoided. In her much-praised recent biography, Brenda Maddox noted that Franklin rarely wore a lead apron and often stepped carelessly in front of a beam. Oswald Avery never won a Nobel Prize either, and was also largely overlooked by posterity, though he did at least have the satisfaction of living just long enough to see his findings vindicated. He died in 1955. Watson and Crick's discovery wasn't actually confirmed until the 1980s. As Crick said in one of his books, it took over 25 years for our model of DNA to go from being only rather plausible to being very plausible, and from there to being virtually certainly correct. Even so, with the structure of DNA understood, progress in genetics was swift. And by 1968, the journal Science could run an article entitled, "That Was the Molecular Biology That Was," suggesting, it hardly seems possible, but it is so, that the work of genetics was nearly at an end. In fact, of course, it was only just beginning. Even now, there is a great deal about DNA that we scarcely understand, not least why so much of it doesn't actually seem to do anything. 97% of your DNA consists of nothing but long stretches of meaningless garble, junk, or non-coding DNA, as biochemists prefer to put it. Only here and there along each strand do you find sections that control and organize vital functions. These are the curious and long elusive genes. Genes are nothing more nor less than instructions to make proteins. This they do with a certain dull fidelity. In this sense, they're rather like the keys of a piano, each playing a single note and nothing else, which is obviously a trifle monotonous. But combine the genes, as you would combine piano keys, and you can create chords and melodies of infinite variety. Put all these genes together and you have, to continue the metaphor, the great symphony of existence known as the human genome. An alternative and more common way to regard the genome is as a kind of instruction manual for the body. Viewed this way, the chromosomes can be imagined as the book's chapters, and the genes as individual instructions for making proteins. The words in which the instructions are written are called codons, and the letters are known as bases. The bases, the letters of the genetic alphabet, consist of the 4 nucleotides mentioned a while back: adenine, thymine, guanine, and cytosine. Despite the importance of what they do, these substances are not made of anything exotic. Guanine, for instance, is the same stuff that abounds in and gives its name to guano. The shape of a DNA molecule, as everyone knows, is rather like a spiral staircase or twisted rope ladder, the famous double helix. The uprights of this structure are made of a type of sugar called deoxyribose, and the whole of the helix is a nucleic acid, hence the name deoxyribonucleic acid. The rungs or steps are formed by 2 bases, joining across the space between them, and they can combine in only two ways. Guanine is always paired with cytosine, and thiamine always with adenine. The order in which these letters appear as you move up or down the ladder constitutes the DNA code. Logging it has been the job of the Human Genome Projects. Now, the particular brilliance of DNA lies in its manner of replication. When it is time to produce a new DNA molecule, the two strands part down the middle like the zip on a jacket, and each half goes off to form a new partnership. Because each nucleotide along a strand pairs up with a specific other nucleotide, each strand serves as a template for the creation of a new matching strand. If you possess just one strand of your own DNA, you could easily enough reconstruct the matching side by working out the necessary partnerships. If the topmost rung on one strand was made of guanine, then you would know that the topmost rung on the matching strand must be cytosine. Work your way down the ladder through all the nucleotide pairings, and eventually you would have the code for a new molecule. That is just what happens in nature, except that nature does it really quickly, in only a matter of seconds, which is quite a feat. Most of the time, our DNA replicates with dutiful accuracy, but just occasionally, about one time in a million, a letter gets into the wrong place. This is known as a single nucleotide polymorphism, or SNP, familiarly known to biochemists as a SNP. Generally, these SNPs are buried in stretches of non-coding DNA and have no detectable consequence for the body, but occasionally they make a difference. They might leave you predisposed to some disease, but equally they might confer some slight advantage, more protective pigmentation, for instance, or increased production of red blood cells for someone living at altitude. Over time, these slight modifications accumulate both in individuals and in populations, contributing to the distinctiveness of both. The balance between accurate